All right, more muscles. Now, I know that last lecture was really long, so this is going to be a little bit briefer. So we're going to talk about smooth and cardiac muscle. And as I said, this chapter really focuses mostly on skeletal muscle, and that's what I want you to focus on. I'm just going to do a brief introduction to smooth and cardiac muscle. So cardiac muscle is going to be something that we'll talk a lot about in 169 when we get to the heart. So cardiac muscle is going to be found in the heart. It has short branching fibers. They each have 1 or 2 nuclei, and it's striated. So it also contains sarcomeres, just like our skeletal muscle. Cardiac muscle has many mitochondria because it uses aerobic respiration pretty much exclusively, and so the heart really needs a lot of oxygen. And it has something called an intercalated discs, which join the ends of neighboring fibers. These contain both desmosomes and gap junctions. So they're like, it snaps the cells together so that they don't come apart, and the gap junctions actually allow fluid and intracellular fluid to be shared between the different cells. Contractions in the heart are started by an autorhythmic pacemaker cells. We'll talk a lot about those in 169. And the heart rate and contraction force are just influenced by the nervous system. So cardiac muscle cells are striated and they are involuntary, and be able to identify them for a lab. They're found in the heart. So we'll talk a lot more about this in 169. Next, we'll move on to smooth muscle. So smooth muscle, this is the only place we really talk about how smooth muscle works. We're going to have a lot of smooth muscle in like the respiratory system, in our GI system, in our vessels, all of this in 169, but I want to mention a little bit about how it works. So smooth muscle is found in many of our organ systems, right? So the blood vessels of the cardiovascular system, it helps regulate blood pressure and flow. In the bronchioles in the respiratory system, it controls the airflow to the alveoli. In the intestines and the digestive system, it mixes and propels material. In the ureters of the urinary system, it propels urine from the kidneys to the bladder. And in the uterus of the female reproductive system, so lots and lots of smooth muscle. We can find it in all of these different areas, and we will talk about the muscle in each different system as we go through 169. So smooth muscle cells are not striated, and because of that, that's because they don't have sarcomeres. They do have actin and myosin, but they're arranged very differently, and so smooth muscle contracts differently too. Smooth muscle cells are much smaller. They have a single nuclei, and they are sort of fusiform in shape, meaning they look a little bit like an ellipse or like a football, kind of. So they're football shaped. They're wide in the middle with tapered ends. They are smaller than skeletal muscle fibers. They also have many different types of calcium channels. So some of them are chemically gated, some of them are voltage gated, some of them respond to the stretch of the muscle, and so they can be activated in many different ways. They don't have T-tubules. So that's different, right? It's weird. They don't have T-tubules. Instead, they have these little things called caveoli, these little invaginations that go in. So they're not quite the same, but it does increase the surface area of the outside of the smooth muscle. And they actually don't have very much sarcoplasmic reticulum because they get most of the calcium from the outside of the cell. So this is what they look like. So we have each our little sort of football-shaped cells. They have a single nuclei, and they have these little indentations called the caveoli. And as you can see, they are not striated. So here we have our actin and myosin filaments, and the myosin is yellow, same as it was before, and the actin is red. And then we have intermediate filaments that kind of attach them to it's like a structural framework that holds them together. And so they look a lot differently. They are attached a lot differently. And right here is called a dense body. And these dense bodies are going to be these little protein areas that are going to attach the actins and the intermediate filaments together. And then the myosins are going to kind of be in between these different areas. And so we have the intermediate filament and the dense body. We have dense bodies in here that are attaching all the actins together. And then we have myosin in the middle. And so when the contraction happens, we're still going to have myosin pulling on actin. So we're still going to have the ends of the actin coming close together. So this is relaxed here and this is contracted here. So we can see how the myosin heads are pulling on actin just like they did before. And when they contract, they actually contract in like this ringing motion because it's not just all parallel myofibrils like we had in the smooth muscle, but you can see they're kind of like crisscrossing across the inside of the cell. And so when the smooth muscle contracts, instead of just getting shorter, it kind of squishes together and it has like a wringing motion, like you were wringing out a mop or wringing out like a washcloth to get all the water out. So it contracts in this wringing motion. And so we also have dense plaques, which are points where the intermediate filaments attach on the inside of the sarcolemma. And so that's because, you know, it doesn't matter if if the thick and thin filaments contract if they weren't connected to the outside of the muscle. So the dense plaques can connect it to the sarcolemma so that the whole muscle is going to contract. As we said, the contractile proteins that act in a myosin are arranged between the dense bodies and the dense plaques. They're oriented at oblique angles to the longitudinal axis of the cell. And that just means instead of being parallel and just shortening in one direction, they're kind of oriented in lots of different areas and when the whole cell gets smaller as it contracts. And we do not have sarcomeres here, and there are no Z-discs, so it's smooth, there are no striations. So how exactly does this happen? Contraction's a little bit more complicated in smooth muscle, or I guess it's not really more complicated, it's just different, and we're not going to talk about it in as much detail, and I might not, I'll ask maybe one question about it, I'm not going to ask that much about how it works, because I want you guys to focus on skeletal muscle. But unlike skeletal muscle, smooth muscle filaments have myosin heads along their entire length, and so they can form additional cross bridges. This is going to allow them to kind of contract more. So even if they're already contracted, they can contract more. Filaments can perform a latch bridge mechanism, so myosin attaches to actin for an extended period of time without using extra ATP, and so therefore they can stay contracted longer without getting fatigued. We'll talk about calmodulin, which is a protein that binds calcium, and we mentioned this in skeletal muscle as we have calmodulin inside the sarcoplasmic reticulum that helps bind calcium, but this is really important in the physiology of muscle contraction in smooth muscle. There is a special enzyme called myosin light chain kinase that's very important. It actually phosphorylates the myosin head when activated by calmodulin, and this is going to be important for activating myosin. And there's another enzyme called myosin light chain protease, myosin light chain phosphatase enzyme that dephosphorylates myosin head, which is required for relaxation. So we'll go through all the steps in just a second. All right, so this is the picture. And so the first thing that happens is that we said there are lots of different calcium channels, and some of them respond to stretch, some of them respond to a nerve impulse, some of them respond to, lots of different things. Some of them respond to voltage, which would be like a nerve cell. But so something, a stimulus, which could be an action potential, it could be stretch, it could be something else, is going to trigger the opening of voltage-gated calcium channels. And calcium is going to enter from the outside of the cell into the inside of the cell. So once again, calcium is really important. But we didn't have to go through nearly as much, as many steps to get calcium into the cell. This is like the first step with smooth muscle. It seems a little bit simpler. So calcium enters the cell. Then calcium enters the cell, it's going to bind to calmodulin. And so calmodulin, remember, is that special protein that binds to calcium. And in smooth muscle, we have it inside the sarcoplasmic reticulum. Calmodulin is then going to form a calcium calmodulin complex, and it kind of changes shape. And that calcium calmodulin complex is then going to activate myosin light chain kinase. This activated myosin light chain kinase is then going to go to myosin. One thing to know is that in this smooth muscle, when you look at the actin and myosin, you don't see any troponin and tropomyosin. It's because they aren't there. There are no chaperone proteins. And the reason we don't need chaperone proteins here is because at rest, myosin is deactivated. So it's a different mechanism of activating and causing contraction. So at rest, myosin is deactivated. It needs to be activated before contraction can happen. So this myosin light chain kinase is actually going to go ahead and activate, it phosphorylates or adds a phosphate to myosin, which activates myosin. And that takes a little while. And so once the calcium enters, calmodulin activates myosin light chain kinase. Myosin light chain kinase then activates myosin. Once myosin is active, there are no chaperone proteins. We don't have troponin and tropomyosin, and so it can just immediately start cross bridge formation. So we're going to have that same shortening where we have the cross bridge formation and all of that, the power stroke and the reattachment. So the activated myosin head is then going to bind to the thin filament to form cross bridges. Myosin ATPase, once again, hydrolyzes the ATP, so it's taking that energy from ATP, and it provides the energy for that power stroke. This process is repeated again, and shortening of the muscle is going to happen. So as I said, it's a different type, it's a different way of regulating skeletal muscle, regulating contraction than skeletal muscle. That's what I just said. So how does it relax? So smooth muscle relaxes by ceasing, so the stimulation ceases. It means we don't have the stimulus anymore and those calcium channels close. The calcium is removed from the sarcoplasm. And then myosin light chain phosphatase is then going to dephosphorylate myosin. So it's going to deactivate myosin. Once myosin is deactivated, then it's not going to form any new cross bridges. But sometimes this can be slow to relax because, remember, myosin can actually stay bound to actin for a pretty long time without that ATP, without extra ATP. So things that how smooth muscle contraction is different from skeletal muscle is that smooth muscle has a long latent period, meaning that it takes a long time for the activation of myosin. So it takes a long time to phosphorylate the myosin head. So even if the calcium gets in pretty quickly, it still takes a while to activate myosin. There's also the ATPase, which is the thing that breaks down the ATP and provides energy for each cross bridge, is slower. There's a long duration. Calcium pumps are slow, so it takes a long time to remove calcium from the inside of the cell. In order to actually cause the muscle to relax, we actually have to have the other enzyme dephosphorylate that myosin head, and that takes a while. And then there's a latch bridge mechanism where actin and myosin stay attached for a long time without additional ATP. And so our smooth muscle can contract really well for a long time. But it's okay that it's slow because it's not something like we weren't, it's involuntary anyway, so we're not worried that it's slow. And it's good that extended contractions can maintain continuous tone, right, in the walls of the blood vessels. So a lot of times we want our blood vessels to be partially contracted all the time. So it's good because we need to have a lot of sustained contractions in our visceral muscle. Smooth muscle is also relatively fatigue resistant, which is good. We don't want our GI tract or our blood vessels to get really tired and stop contracting. The energy requirements are really fairly low compared to skeletal muscle, and once again, that latch bridge mechanism makes it easier to maintain the contraction without spending additional ATP. And that length tension curve is actually really broad because this allows the muscle to contract very effectively when it's stretched out or when it's contracted. So just the way that because we don't have sarcomeres, we don't have Z discs, the way the thick and thin filaments are arranged makes it very efficient. And so it can contract forcefully even if it's really contracted or really stretched. So 50 to 200% of resting length. So it allows emptying of the bladder even if the bladder is really stretched or if the bladder is really small, which is kind of important. So there's a couple of different ways that smooth muscle can be controlled. So the first one is that it's involuntary. So the autonomic nervous system can control it. That's the involuntary part of our nervous system. We'll learn about it in chapter 15. And so the muscle's response depends on the neurotransmitters present and the muscle's receptor for it. So it gets a little bit complicated, but our autonomic nervous system can tell it to contract or relax. So for example, smooth muscle of the bronchiole contracts in response to acetylcholine and relaxes in response to norepinephrine. So different neurotransmitters can cause a different result from the smooth muscle. There's also a really important one that these muscles often respond to stretch. So the myogenic response is a feature of smooth muscle where a lot of times it contracts in reaction to stretch. So when the muscle is stretched, it it like reflexively contracts. That's a reflex. And we'll talk about the myogenic response in 169. The Streck's relaxation response is relaxation after a prolonged stretch. So we've got a couple of different like automatic reflexes of smooth muscle. And then other stimulating factors could include various hormones like low pH, low oxygen, high CO2, certain drugs, pacemaker cells. So there's lots of things we'll talk about in 169 that are going to cause reflexive contraction or relaxation of those smooth muscle. So now there's two functional categories of smooth muscle, multi-unit smooth muscle and single unit smooth muscle. So multi-unit smooth muscle is going to be pretty similar to skeletal muscle. So it's arranged in units that receive stimulation to contract individually. So it's found in just a few places, in the iris and ciliary muscles in the eye, erector pili muscles in the skin, larger air passage in the respiratory system, and the walls of the larger arteries. And so in this case, it's a lot like skeletal muscle. The degree of contraction depends on the number of motor units activated. So it's very similar to skeletal muscle. And so we'll look at this picture here. So single unit smooth muscle, multi-unit smooth muscle, where we have one motor neuron, right? And then it's going to be innervating, a whole motor unit. So a motor unit, remember, is one motor neuron and all the muscle cells it controls. And so this one is going to, control all of these muscle cells. And then we might have another motor unit that controls all of other muscle cells. And so it contracts. In order to contract more, it needs to recruit more motor units. It's a lot like skeletal muscle. Single unit or visceral smooth muscle is the most common type, and this is a little different. And in these cases, we have, these areas of smooth muscle are all connected by gap junctions, and that means that we have little tiny holes in between each cell that allows ions to move from one cell to the next. And so as the ions move from one cell to the next, the action potential can just immediately move from one cell to the next. And so all of these cells can contract in unison because they're linked by these gap junctions. And so usually they'll form 2 or 3 sheets in the walls of a hollow organ. And we'll talk a lot more about this in 169. So the walls of the digestive, urinary, and reproductive tracts, portions of the respiratory tract, and most of our blood vessels. And so how do these get stimulated? So we have a nerve, an autonomic nerve, and this nerve has something called a varicosity. So the nerve has all these little swellings along the nerve. And these swellings contain synaptic vesicles of one type of neurotransmitter, either ACH or norepinephrine, depending on what type of nerve it is. And receptors are scattered across the sarcolemma of these cells. And so you can see right here, this is the nerve, and it has these little areas. These are called varicosities. It's almost like a bunch of synaptic knobs in a row, where we have synaptic vesicles here. And once these synaptic vesicles are released, they're going to go and activate this smooth muscle. And so these different smooth muscles here, the ones that are right next to the varicosities, are going to be activated by the nerve. But the other ones, so each of these muscles are connected by gap junctions to all the rest of the muscles. And so as the action potential moves through one cell, it is automatically transmitted to the next cell, and the next cell, and the next cell, and the next cell. And so they can all contract as a unit. All right, so this is really useful that they can contract as a unit so that, all of your digestive tracts and your peristalsis and things, as you move through the digestive tract, it's very well coordinated. So this one I know is a lot shorter, so hopefully that is good. I do want you to know a little bit about the different types of smooth muscle and a little bit about smooth muscle contraction, but focus mostly on skeletal muscle.